Okay, so we're going to talk now about cytokines in the immune system, and some of this is going to be a review about what we've already talked about from previous lectures. So we have, of course, our innate and our adaptive immune response, and cytokines are really soluble messengers that allow the immune cells to communicate. Now, some cytokines very rarely can be cell-associated, like TNF, but most are going to be released. And what's important is most are released after activation. There are some cytokines that are going to be constitutive, and those are mostly going to be growth. So, for example, we have our TNF, and we have our interferons, type 1, which is our alpha, beta. And these are going to be secreted by immune and non-immune cells. And then we have some constitutive cytokines. We have IL-7, and we have, which is going to be for the development, if you remember, of our B and T cells as they're going through the primary lymphoid organ. Plus, it's a growth factor for naive T cells. So as the naive T cells are circulating for their 30 days, as long as they keep seeing IL-7, they will keep going. And then we also have IL-15, and this is going to be a growth factor for memory cells. And again, these two are going to be more constitutive. But a lot of the cytokines we're going to talk about are the ones that are made after they get exposed. So your innate response will release cytokines that will communicate with your adaptive response, that will release cytokines. And all of these are trying to get both your innate and adaptive activated so they can go after whatever is the threat. So cytokines have a few features. One is that they're redundant. So cytokines can have similar properties. So an example of this is going to be IL-1 and TNF. They work in synergy, and the impact of both is going to be more than the impact of one alone during the acute phase response. APR is acute phase response. Pleiotropism or pleiotropism is that many cytokines affect several different types of cells. Okay, and so we're going to have different types of secretion. So we have immune-only cytokines, and this is typically going to only follow activation. And this is tightly controlled, so only the cells activated will release the cytokines. Non-immune can be cytokines such as TNF-alpha. We have interferon 1, not interferon 2. Interferon 2 is interferon gamma, that is immune only. I'll put interferon 2 up here. And we typically write it, I should write it with Roman numerals. It's typically written by numerals. We have constitutive, so that's going to be things like our IL-7 and our IL-15 that we just discussed. IL-7 more for development and naive, IL-15 more for memory cells. We have inducible or transient, and these are again going to be followed by activation. And it's actually, they're going to be at low levels. So they typically work locally. They're not going to enter the blood and work at other things unless they're made too much. So you have too much inflammation, then you can start to see these things pop into the blood. Now, control of the secretion can be autocrine. And so an example of this is IL-2. T cells will make IL-2, and then IL-2 will feed back on the same T cell, or paracrine. IL-2 is another example of this. So T cell makes IL-2, its autocrine also helps or works with the neighboring cells and can be paracrine. We have endocrine or hormone-like, and this is going to be more of the innate cytokines. Our acute phase, our IL-1, TNF, alpha, and our IL-6. And so our autocrine or our paracrine could also be innate. It doesn't always have to be adaptive. IL-2 is from the adaptive. But it can also be below into this one. Macrophages make IL-8, which is a recruitment of newts. So that's another example. We can also have other cytokines that are going to be made locally in a paracrine. So this is another one, is going to be inhibitory. So that can be IL-10 or the TGF-beta. But the endocrine-like are going to be more of those innate. So as a side note for our type 1 interferons, so for hepatitis C and B patients, type 1 interferon was a treatment with an antiviral rabdovirin. And so they had side effects. Because type 1 interferon, of course, can inhibit protein synthesis. So that would not only inhibit protein synthesis of virus-infected cells, but also other infected cells. It also can initiate the acute phase response. It's an APR, acute phase response. And so the hypothalamus has receptors for type 1 interferon, so it can cause fever and things like that. And to date, you might hear terms like pegylated interferon, and the PEG is just going to make interferon more stable, so it has a longer half-life in the body. But antivirals have really replaced most of the type 1 interferon. In developed countries. But in developing countries, interferon might still be used to treat hepatitis infections. Okay, so here's a nice little table about all these different lovely cytokines, and we've talked about most of these, so this should be very familiar. On this table, just to point out a few things, we have our pro-inflammatory cytokines, so those are TNF, We have IL-1 or acute phase response. We do lump in chemokines, which are going to be directional. IL-8 is also a directional, though it's not a chemokine. Chemokine have or CX, CR type of designation. So if you see CXCR4, CCR4, etc., those are types of chemokines. Type 1 interferon, which is going to be more antiviral, intracellular pathogen. IL-6, one of our acute phase responses again. We have our growth factors. So our growth factors are IL-7, which is going to be important for our progenitor cells and our naive T cells. We have granulocyte colony stimulating factor for our neutrophils. Remember that macrophages are made at a steady state. Neutrophils are going to be increased when there is an infection, not in the macrophages. IL-2 is a growth factor for T cells, a very important growth factor we'll talk a little bit more about in a minute. We also have our different T cell subsets, CD4s. These are CD4 subsets. Th1 and Th2 kind of inhibit each other. So if your body does decide to go with a Th2 response, it'll suppress a Th1 response and vice versa. So it's important to get those correct, or else you have the wrong response for the wrong potential pathogen. Our Th1s are going to be interferon gamma, IL-12. Our Th2s are going to be IL-4, IL-5. 17 is going to be 23 and 17. And then we have, of course, our Tregs, IL-10, TGF-beta. And so all of those are important to know. The types of cells that make them, hopefully you know what they are because of previous talks about them, and also what they're trying to target. So our Th2 are trying to go after our eosinophils, mast cells. Our Th1s are trying to get our CD8 T cells, NK cells activated. Th17 is for neutrophils, for our extracellular bacteria to eat them. And Tregs, of course, can suppress all of them. Okay, so let's talk a little bit about receptors. So we know cytokines get secreted, and so they have to be interacting with some receptor on a cell, just like anything else that's secreted. Hormones they use the same thing, receptors. And so one receptor that's important to talk about is the common cytokine receptor families. So those are over here. So we have a gamma chain. And this is important, the common gamma chain. So you can see that the gamma chain is shared between IL-2, IL-4, and IL-7. And so just to show what this means, if you have a T cell, it will have the gamma chain on its surface, okay? And if this T cell is going to become a Th2, the gamma chain will be joined by the alpha chain for IL-4. If this is going to become Th1, you will have the gamma, beta, alpha for IL-2. And so what's common, though, is the gamma. That doesn't change. So that's called the common gamma chain. And so people who have deficiencies of this gamma chain have a disease called severe combined immunodeficiency, which is X-linked. And that's because they can't respond to any of these cytokines. They all share the same chain, and so they're not able to respond to this. Now, the the common cytokine chains, another thing to point out is that they are going to use that JAK-STAT signaling. Get rid of this. It's good. They use JAK-STAT. So remember, JAK-STAT is the Janus kinase signaling system. And STAT is the signal transducer and activator of transcription. So JAK is associated with the receptor, and your stat becomes the signal. So for example, here we have a Th1 secretes IL-2, right? And your CTL. You have on the surface what's referred to as a high affinity IL-2 receptor. And this is that gamma chain and beta chain. And these guys are constitutive. So gamma plus beta plus alpha is the high affinity. And the alpha for the IL-2 equals CD25. So that alpha chain on the IL-2 receptor is also referred to as CD25, and it's the high affinity. And so this is going to lead to activation of JAK. And then JAK is going to activate STAT, remember as a dimer, and that will go to genes that are going to be expressed. So nothing different than what you already know. Just a little bit more information. IL-2 is going to activate JAK5, STAT1, and STAT3, for example. And so what that means is that we have a lot of different JAKs and STATs. And for our T cells that are responding to IL-2, it's going to activate these particular JAKs and STATs, and IL-2 is a growth factor. So one thing that IL-2 does, it increases BCL2, which remember is anti-apoptotic. Okay, so it's going to keep these cells alive, the signaling with the IL-2. It's also going to cause them to replicate. So genes necessary for replication are also going to be activated through the transcription factors STAT1 and STAT3. Okay, so now let's just talk very briefly about chemokine. Chemokine receptors use our G protein receptors. Those are 7 transmembrane receptors, similar to epinephrine, glucagon, those type of receptors. They are going to be involved in leukocyte migration. And they have nomenclatures such as CCL14, CCL19, CCL20, CCL25. We have CCL27, CXCL12, CXCL13. Oops. We have HIV that uses chemokine receptors as co-receptors, such as CCR5 is such a receptor. And so basically, chemokines are on the surface of your white blood cells, and they give them sort of an address. Where do you go in the body? Are you going to go to the mucosal system? Are you going to stay in the peripheral system? Do you need to go into the lungs? Do you need to go into the liver, etc.? So it kind of gives them an address to go. And what's really interesting is that what we do know is that your memory cells remember where they went. So if you had a gut infection, your memory CD4 T cells will, if you get that second gut infection again, will remember I was in the gut. So they will go very quickly to the gut. And that's because of the chemokines. They're going to have the right chemokine to get them into the right location. All right, so let's talk a little bit about something called receptors and families. So we have things called superfamilies. So TNF superfamily, receptor superfamily. So the TNF receptor has a cytoplasmic tail, which has a death domain, and it can activate caspases. So another member of the TNF members are CD40 and Fas ligand. So those are listed down here. So they're all part of the TNF family of receptors. So TNF and Fas are going to have the death domain. And this is in the cytoplasmic. And this can activate your caspases. Okay, So a TNF can induce apoptosis. But it doesn't always. For example, this has to be in an infected cell. And in a macrophage, TNF in a macrophage will induce NF-kappa B, that transcription factor. And that will turn on certain genes. Our Fas ligand interaction will induce apoptosis. Our CD40 ligand will induce HELP. And the HELP will be different if it's a B cell or a macrophage. So you can have CD4 B cell HELP, and you can have CD4 macrophage HELP. And this will be for isotype switching, plus hypermutation, somatic hypermutation. And remember, isotype switching is going to depend on Th1 versus Th2 versus Th17. So you'll get IgG. Oops. Just doing a little review here. Th1 will drive IgG responses, Th2 will drive IgE responses, and Th17 will be IgG responses. And so the CD40 ligand interaction is very important. And so if you have mutations in that ligand, which is also known as CD154 mutations, then you have antibody deficiency. Okay, so on your table here, you can see these are TNF superfamily, and the TNF receptors are going to be CD40 as well as Fas. So you can see that whether the signaling transduced by these receptors induces apoptosis, yes for TNF and yes for Fas. And whether they induce transcription, it's yes for TNF and yes for CD40. Because remember, TNF induces transcription in our macrophages. So TNF can do both. Fas really is just for inducing apoptosis, and CD40 is just for helping. It does not induce apoptosis. Now, so the role of the cytokines in our immune response is very variable. So we have many different roles. We're going to have the role in inflammation that we've been talking about. We have a role of activating. We have a role of skewing. And we have a role, of course, in contraction of a response. So we basically have, after an infection, your macrophages really quickly, very quickly will make these early cytokines. So again, we have our acute phase response. We have our cytokines that's going to help drive neutrophils, neutrophil growth. And then we have later in an infection, the antigen-presenting cell will be secreting other cytokines such as IL-1 and interferons. Interferons, remember, are plasmacytoid. Dendritic cell makes a lot of type 1 interferon. It's a major producer of type 1 interferons. On activation, our T cells are going to secrete IL-2, which is a growth factor for other T cells. And it depends on what type of infection your T cells have as to what type of response. So your T helper cell is kind of the orchestrator of your responses. You can have a Th1 cell for an intracellular pathogen, which will activate your CD8 T cells to kill infected cells. You can have a Th2 response if you have a parasitic infection, which will activate your mast cells. You can also have an extracellular bacteria infection, which will have a Th17 response, as we've been discussing. And keep in mind that your anywhere where you have an infection in your gut, you're always going to have a predisposition towards tolerance, and so your TGF beta is going to be more prominent in those locations, and you're going to get an IgA response instead of an IgE. IgA is secreted into the lumen of the gut, so it doesn't really promote inflammation in the gut proper, in your cells, inside your body. It really is there to help neutralize pathogens extracellularly. Whereas if you had an IgG response, that would actually induce inflammation in your gut. So you want to kind of avoid that. Now let's talk about cytokines and acute inflammation. So let's talk about our story. We have our bacteria, and they have their PAMPs, pathogen-associated molecular patterns. And this is going to activate our innate response. Via the pattern recognition receptors, such as our toll-like receptors. And then these are going to release cytokines. We have our acute phase, IL-1, IL-6, TNF-alpha. We have our granulocyte colony stimulating factor for newts. We have our IL-8 to recruit newts. And these are going to give our acute phase, which will be our fever, increase white blood cells, recruitment, decrease our appetite, increase malaise. All of this can lead to septic shock if it's not under control. And then we're also going to have local inflammation. We're going to have our IL-8 and chemokines, which will recruit newts. We can also have IL-23, which will be for a Th17 response. And then we can release IL-17, and that will attract neutrophils as well. This IL-17 will also stimulate epithelial cells to release antimicrobial peptides. Which are defensins. And defensins work the same way as MAC does. These will punch holes in bacteria. Now, if viral, which is intracellular, or an intracellular bacteria, the cytokines would favor Th1. And so you would get your IL-12, which will stimulate NK cells to release interferon gamma, etc. Okay? But it's the same thing. Your macrophage will get activated by those pattern recognition receptors for viruses, which will be associated with double-stranded RNA, things like that, which will lead to a Th1 response versus a Th17 response. Okay, so cytokines are what are going to generate the right response via your CD4 becoming the right helper, because that right helper is really what's going to drive. And so this all is going to occur in the T cell zone of the secondary lymphoid tissue. So this would be your lymph node, the white pulp of your spleen, things like that. So the DCs are going to be what activates your T cells. So DCs are going to be activated at tissue with the danger signal. Danger. And this is going to increase CD80, 86, also called beta 7, and it's going to increase ICAM. Now remember, ICAM is intracellular adhesion molecules. And ICAM1, remember, is on our white blood cells. ICAM2 can be on your endothelial cells. VCAM, is vascular cell adhesion molecule. Okay, so your DC is coming in. It has its adhesion molecule. It has its second messenger or second signal, the CD80. It can be also secreting cytokines. It can be secreting IL-1, interferons. The T cell becoming activated will express CD25, which is the alpha chain of the IL-2 receptor. And so it's going to have the high affinity alpha-2 receptor. Which includes gamma, beta, and alpha. And alpha, again, is CD25. You might see CD25. It's going to start secreting IL-2. And it's going to work autocrine and paracrine. And then it's going to proliferate via IL-2 signaling. So IL-2, of course, is very important for activation. And then the T cell has to decide, am I going to become a Treg, Th17, Th1, or Th2? So let's talk a little bit about these. So the first scenario we have is in the gut. And we have our epithelial cells. And they are going to be producing TGF-beta to try to create sort of that tolerizing environment. And this is going to cause a CD4 to become an induced Treg. This is also going to cause a B cell to switch from IgM to IgA. IgA skewing is important. To protect the lumen from pathogens. And then also we have our Tregs, our regulators, are important to decrease inflammation in your gut to antigens that you don't want to respond to, for example. Our second scenario is our extracellular pathogens. And in this case, you want a Th17. And so you have your CD4 T cells, and your innate cells are making IL-13. And this is going to drive your CD4 T cells to become Th17. And they are going to release IL-17, which recruits newts. And then we have our intracellular pathogens. Which is the most abundant because of viruses. Viruses are our most common pathogens. And so for this one, of course, you want a Th1 response. And so in this case, you have your macrophage produces IL-12. This activates your NK cell. Which then produces your interferon gamma. And then your interferon gamma and IL-12 will drive a Th1, creates that environment for a Th1. This will express Tbet, which is a transcription factor for your Th1. You're going to release interferon gamma, and this is going to cause your IgM to switch to IgG for your B cells. In a chronic state, you also have TNF, and in a chronic, you get granulomas. The next one is a parasite, worm infection. This is a Th2, and so you have your T cell and some signal that's not quite understood. It might be your basophils. It could be that you just don't have IL-12 and interferon gamma, so you default back to a Th2. But whatever it is, you get your Th2 cells. This has GATA3 as a transcription factor. And then this is going to lead to IL-4 release. And this is going to cause your switch to IgE. You also get IL-5 release from a Th2, and IL-5 plus eotaxin is going to recruit mast cells and eosinophils. And of course, IgE will play a role in this too, and mast cells will make IL-4. So they'll continue to drive this Th2 response. The mast cells will once they become activated. And then finally, we have also, just with respect to our Tregs, you can have natural Treg, which is from the thymus, or you can have inducible Tregs, which is from the periphery. They're both going to have FOXP3 as a transcription factor. And so what I want to point out here is FOXP3 deficiencies is your life is about 1 year, you die. Okay, so again, peripheral tolerance is really important. You can't survive without peripheral tolerance. Cytokines are going to be our IL-10 and TGF-beta. Okay, so now let's talk a little bit about contraction. So again, in an immune response, this is our phases. And we'll say T cells. Okay, so our phases of T cells are going to be activation, expansion, contraction, and then memory. So this is activation. This is going to be expansion. This is IL-2 driven for T cells. We have contraction. And then we have our memory. Now we're going to talk a little bit about contraction. So, step 1, pathogen is cleared. Right? And so you lose, when the pathogen is cleared, this is going to result in decrease of your cytokines. It's going to lead a decrease in antigen presentation. It's going to lead to a decrease of IL-2, a decrease of CD25, which is the high affinity IL-2 alpha receptor portion, a decrease of BCL2 because you lost IL-2 signaling. So you can't maintain that high BCL2, and it's going to lead to an increase of apoptosis. And that equals contraction. And then finally, your memory cells will survive. Now, is it about location? Do your memory cells survive because they just happen to be where they saw IL-15 and they outcompeted the others? There's not enough IL-15 to go around, so only those that see it are able to survive. And that's still under investigation, exactly who becomes memory and who dies. But for the purposes of this, for contraction, this is going to be driven by apoptosis because you lose the survival signals primarily through the IL-2, through the IL-2 high affinity receptor CD25 portion, and then you lose your BCL2 expression. Now, some clinical uses of cytokines, let's just talk briefly about them. Keep in mind, normal is usually localized. So you have autocrine and paracrine. An exception is our acute phase. And during shocks, when you have too much around. So some clinical uses are recombinant IFN-1 treatment, and this is for Hep B or C. And also we use monoclonal antibodies to block cytokines. And there's cases in the book that clearly just show this. But basically, you can have, for example, anti-TNF alpha antibodies for rheumatoid arthritis. As an example. In general, monoclonal antibodies in a lab can be made against any antigen. So we can make monoclonal antibodies to our cytokines because cytokines are encoded for by us and they are in fact antigens. You have to be very careful when you do things like this because of course they can lead to side effects, can lead to immune suppression, be susceptible to other infections, etc. So they're used they're very controlled in their use, but they can be made. Okay, so that is the end of cytokines, and thank you for listening.